Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Nazish Bhatt, moderator of the research workshop organized by Pakistan Society of Hepatology. So I welcome you all in this research workshop. So the theme of this research workshop is pursuing research in clinical hepatology. As you all know that currently research is the integral part of our all postgraduate training and our the career in hepatology and gastroenterology. So it's very important for the undergraduates and postgraduates and the consultants to learn about the research. So today, the facilitator of this workshop is Professor Mohammad Saleh, and he is a renowned professor and transplant physician currently working in Qazi-Azam Hospital, Islamabad. And we have chairpersons. I welcome all the chairpersons. Professor Akhtar Shiri from Kohat, Professor Khalid Mahmood from Peshawar, Professor Shahab Abid from Aga Khan University Hospital, Karachi, Professor Faisal Wasim Ismail, Aga Khan University Hospital, Karachi. So before I start the talks, there is pre-assessment pool which will be done by Dr. Shahid Sarwar. So Dr. Shahid Sarwar, are you listening me?
Bismillahirrahmanirrahim. Can you hear me?
Yes.
Yes, I'm listening.
Walaikum salam.
Should I start?
Yes.
So let me
Yes,
share.
please.
Can you see my desktop?
Yes, Dr. Shahid.
Okay. Bismillahirrahmanirrahim. I'm extremely thankful to Pakistan Society of Hepatology for giving me opportunity to be here with all of you. My job in next 10 minutes will be to briefly introduce why are we conducting this workshop.
And research in hepatology, and I would like to invite Professor Adil Ajwad Bhatt. Professor Adil Ajwad Bhatt is Professor of Medicine, Professor of Healthcare Policy and Research at Weill Cornell Medical College, New York and Qatar. Professor Adil Ajwad Bhatt.
Good morning and Assalamu alaikum. I'm very grateful to the organizing committee and the scientific committee of the Pakistan Society of Hepatology for inviting me to give these talks this morning. My first talk is about how to plan research in hepatology, and I have no conflicts of interest to declare about this talk. The outline of the talk is that we will talk about the things that you need to do before starting a research project, talk about individual components of a research project and the details of each one of those. And these includes aims and hypotheses, background and significance, your research strategy or methods, and ethical and regulatory considerations. We will talk about some common clinical study types and how to execute a good research project. And I must preempt that we will talk mostly about clinical research studies and not bench studies or lab studies for today's talk. So how do you develop a research project? I think any good research study or project begins by asking a clinically relevant question. And this question can come from your clinical practice where you see a patient and are looking for an answer but cannot find one, or by your own reading. And again, when you're reading and looking for an answer and that answer is not readily available. Once you have that question in mind, you need to do a good and thorough literature search because it is quite possible that one or many people may have already answered that question. If after a thorough literature search that question is still unanswered in your mind, then you can develop that into your own research project to answer it. And the first step towards that would be developing specific aims and hypotheses that you would test and achieve by doing this project. Once you've developed an idea of what specific aims and hypotheses that you're going to work on, you develop or gather the study tools to design that study. That includes determining what statistical analysis you will use, what sample size will be needed, and power calculations to make sure that your study is powered enough to answer that hypothesis in a scientifically rigorous manner. And of course, never forget the regulatory and reporting requirements, which are very important in any clinical research project. Clinical questions can be one of many. It could be about trying to ascertain the presence or absence of disease, trying to determine how accurate a diagnostic test or procedure is, determining the frequency or rates of disease, risk factors associated with a particular disease, how a treatment affects the disease course, clinical outcomes associated with that, and how can you prevent a disease from happening in the first place. And not the least of which is determining the cost associated with the disease burden. Outcomes can be one of many, depending on the specific question that you're asking, but I categorize them into six major categories and I call them the 6Ds. Death is the hardest, strongest outcome of any clinical study, but you could also look at presence or absence of disease or its progression, the discomfort or disability associated with the disease, the dissatisfaction that the disease causes, and the disparities in diagnosis, management, and outcomes of diseases in one population versus another. As I mentioned before you even start the study, you need to know your field very well. You need to do thorough literature search and reading, talk to colleagues, mentors, other potential collaborators. Think about your patients. What are the kind of things that would be important when you're actually translating your study findings or the answers that you find into helping your patients that you see? Some of the resources that you can look into, tap into to do this kind of knowing your field exercise are PubMed, NIH Reporter. Uh, there are public websites or repositories where every clinical trial has to be registered. Example of that is clinicaltrials.gov. And you also should look at society meeting and abstract books because preliminary data are often presented at these meetings before it gets published in major journals. Before you start any research project, once again, think of the relevance of the project to your clinical practice and how that would affect your patients. Think of availability of resources that are needed to execute this project, the time you'll need to devote to this, the logistics, the collaborators that you need, team members that you need in order uh, to do this project properly. Try to get copies of other projects which are similar to yours, if at all you're able to, and that will help you guide through this process. And each step of the way, think yourself as a critical reviewer and critique yourself honestly and carefully to make sure that you address all the possible pitfalls. Now, often a time you would be applying for funding to conduct a research project. And there are several well-defined components of a research grant application. We will concentrate today on the research plan itself and not go into the details of some of the other things for the lack of time. So let's concentrate on the research plan. The four major components of a research plan. This is what we call the meat of the grant or meat of your whole exercise. The research plan consists of background and significance, aims and hypotheses, your research strategy, and ethical consideration and regulation that you have to follow. Each component has subcomponents which will require more details that you will need to provide. And templates may vary when you're putting together a research project, but I strongly advise you to develop a project template or a clinical research study template so that you don't miss anything when you're designing a project. Let's start with hypotheses and specific aims. You generally start with a broad idea of what you want to understand or answer through your study. Once you've had that broad idea, you develop that into a more focused hypothesis. And hypothesis is a statement which is statistically and scientifically testable. So you can either accept or refute a hypothesis based on the data that you generate and analyze. You recheck the recent literature to see what has been done and what is being done in that field to make sure nothing has been added to the literature from the time you had the original idea at the beginning and now that you develop these hypotheses and specific aims. Develop an outline of what you want to achieve and the specific hypothesis that you want to test. I often get asked the difference between hypothesis versus specific aim. Specific aim is what you want to achieve through a proposal. An example of that would be a statement like, our specific aim is to determine the efficacy of agent A for any condition Y and its impact on outcomes. And the hypothesis is the question or statement that you will test for the truth of that aim. And it is a statistically and scientifically testable statement. An example in this case would be that treatment A will achieve higher rate of cure than treatment B. Now, this statement you can actually test uh, by using statistics on the data that you generate. This is another example of hypothesis versus specific aims, but I won't go into the details of that because I think you get the idea. Now, what are the attributes of a successful hypothesis? I think one of the most important things is that it has to be novel or new. If 50 or 100 or 1,000 people have already answered that question in different ways, then very little can be added to that particular field of study, and not many people will be willing or eager to publish that study. So think about something that's novel, that has not been answered before. It has to be significant to your patient population or your clinical practice or someone's clinical practice. It has to be relevant in the context of where you practice and the kind of work that you do. Make sure it's focused so that you can properly test that in a scientifically rigorous manner. It also has to be plausible. In other words, you can't throw in a hypothesis that has absolutely no basis in truth or common plausibility. And last but not the least, it should be achievable. Don't come up with hypotheses that you will never be able to achieve, no matter what resources you're given and how much time you have to prove that. Then when you're putting these together, what you do is you answer some of these questions. And why is this topic so important? And again, I think it's important to answer these upfront because you'll need that throughout your project execution, as well as when you're writing the final reports and the final paper that comes out of it. So why is this topic so important? What are the critical gaps in knowledge? And how does your project bridge those gaps in knowledge? How will you actually achieve your desired outcomes? And here's where your strategy and methods come in. And then you go on to list your explicit hypotheses and aims. I often find it useful to have a conceptual model so that it can guide me in a visual format how to proceed through this process. And a conceptual model is nothing but starting with your question or your target population on one end and your outcomes at the other end, and try to determine all the mediators, moderators, and factors that impact the outcomes from the exposure or from the disease or the patient population that you start with. And again, I've given one example out here, and arrows tell you that all these things or all these factors mediate the outcome, which is listed on the right-hand side of the slide. Next part is the background and significance. It is important to list that for a couple of reasons. One is that if you're actually writing a budget proposal or a grant application, this becomes a critical piece of that. Even if you're not doing writing a grant application, this part is important because this can form the basis of your introduction to your paper that you'll write after you've gathered all the results and analyzed them. Highlight what is novel and innovative in the significance part. How will your project increase the knowledge in the field? How will it transform the care of patients in the short and long term? And relate it to the bigger picture of betterment of individual as well as population health. Next part of this is preliminary data. And how does preliminary data help you? It helps you in four different ways, at least in four different ways. Preliminary data is a critical building block to support the hypothesis that you're generating. And your preliminary data is actually what it is: preliminary. It has not answered your hypothesis definitively, because if it did, then you wouldn't need to do the study. It provides background and context to what you're trying to do so that people who are going to help you or who are going to critique your project, whether in a grant application or in a manuscript, they understand the context of the whole issue. It helps you determine sample size and power because you will need some of the assumptions to calculate those. It also helps you identify potential pitfalls that you may face when you are executing or implementing this project. The chunk of this exercise relates to your research design and methods. This section, of course, will vary greatly with the type of studies you're proposing, but sufficient details need to be there regardless of what you're proposing. You have to organize your thoughts and your methods along the hypothesis in a logical and sequential manner, and need to identify alternative approaches as well. Here are some of the subcomponents or subheadings that you need to put into your research design and methods or your research strategy. You need to determine what study population you're going to study. How will you recruit patients, including what will be the inclusion and exclusion criteria? What data collection tools and methods you will use? What will be the definitions of every single variable that you're going to collect in terms of data? And for logistics, who will do what and how will they do it? You have to have a team that has enough expertise and time that they can devote to your project. Then you will determine the type of statistical analysis that you will be performing. That includes summary statistics, the types of modeling, and overall methodology. And then you will go into specific hypothesis testing, that how will you scientifically and statistically test each of the hypotheses that you put forward. It is critical to put in a section about how you determine your sample size, or if you have a defined sample size, what will be the power that you will have to detect the difference that you're expecting to find in the various groups that you're studying. And then you put in your limitations and risk mitigation approaches in this as well. There are four major types of, or three or four major types of clinical studies that we often do. These are case control studies, cohort studies, which are non-intervention and observational studies. And then you have intervention studies, one type of which are clinical trials. Now, case control and cohorts are both, as I said, observational studies of risk factors and outcomes. However, they differ in the design. Intervention clinical trials are, as the name suggests, studies in which you institute some sort of an intervention to one group, or more if you have a control group as well. And then you test that. This intervention could be a drug, it could be a device, or it could be any other behavioral, psychosocial, or other intervention. I ideally would have liked to go into details of types of studies, but I'll shorten that for the sake of time, but we'll be happy to answer any questions around that. So what I'll do is discuss some advantages and disadvantages of case control studies, cohort studies, and clinical trials. So advantages of case control studies are they're good for uncommon or rare diseases. You do not need to wait for a long period of time for the outcome or the disease to develop. So in that way, it can help in determining causality in such cases in less time and with much less expense than, for example, clinical trials. The disadvantages are that certain biases can be introduced because you're not doing a controlled or a randomized trial. Investigators, which will be you create a comparison group for the study. And this is where some sorts of selection bias or confounding bias can creep in. To mitigate that, you should select cases and controls from the same base population, who should otherwise have similar baseline characteristics, except the fact that they have different disease that you're studying. You can also use match controls to limit that bias. For cohort studies, the advantages are that you can establish true risk associated with the risk factor over time. The same logical as the clinical question, that if exposed, does a person get the disease? This is what cohort studies answer. You can assess the relationship between exposure and many diseases, because if you follow cohorts over a period of time, whatever outcomes they develop, you can study them. The disadvantages of cohort studies are they're inefficient for uncommon events or uncommon diseases. They are somewhat more expensive. They require a longer duration for the results to be available. Assessing the relationship between the disease and relatively few risk factors, which are defined at the beginning of the study. So you can't add on things later on once you've defined the cohorts initially, because then you won't have those data for all your subjects that you included at the beginning. Clinical trials, of course, are the gold standard for determining effect of interventions. In these type of studies, you can minimize bias by random allocation or random assignment of patients to one or the other group, and they can actually determine causality with relative precision. The disadvantages are that they're very costly, they require large numbers, and they may need to run for long durations of time. You may also have difficulty in enrolling patients in clinical trials, depending on various factors. In research design and methods, other things to consider are missing data, particularly when you're doing observational studies or retrospective studies. This is a major problem, and you need to address that upfront. How would you address missing data? Attrition of study subjects, in other words, a patient may be lost to follow-up. You need to determine other limitations of your approach and identify alternative approaches. One thing that people often do not pay enough attention to is the appropriate expertise required to conduct a study and appropriate manpower to execute that project. This includes the principal investigators, which probably often will be you, your co-investigators, collaborators, consultants, and support staff. Each of these persons has a unique role to fill in a study, and that should be defined clearly. But each person should add value and critical expertise to your study. You basically need diverse expertise in all areas of the project so that people can pool their knowledge and come up with a good, coherent plan. You need to have subject matter experts, study design experts, study coordinators, statisticians, regulatory experts, and others depending on the type of study that you do. And don't forget that for any good study, you may require money and resources. Requirements may vary depending on the scope of your project. However, you need to justify each and every item that you're going to ask money for. And you need to include all your direct and indirect costs when you're putting a budget together. And make sure you ask for enough to carry out the project. Don't skimper on this time. And that includes travel costs associated with presenting your results, publication costs, and any other costs that may be added on later. Finally, you need human subjects protection section. This requires obtaining your institutional approvals before you start any research project. If you are enrolling subjects, you need to have their consent. If it's a clinical trial, you need to have a DSMB or Data Safety Monitoring Board and stopping rules in case of if you find early differences in the two groups that you're studying. So in summary, if you have to put this together, these are the steps that you require to develop a good research protocol. You have to clearly define your question that you're asking, need to know your field in depth to understand what the critical gaps are, determine the resources and logistics that are needed to carry out and implement the project, lay out your complete overall strategy in detail, obtain all necessary approvals, and then you can start this project and take it to fruition. I'll stop here and will be happy to answer any questions that you may have. Thank you for your patience and listening.
Talk. So I would like to mention that Dr. Adil Ajwad Bhatt is a recipient of distinguished PSH awards in academics. Thank you, Dr. Adil. So our next talk is on guidance for statistical analysis and research, and I would like to invite Professor Khalid Mumtaz. Dr. Khalid Mumtaz is Associate Professor at the Division of Gastroenterology, Hepatology, Nutrition at Ohio State University, Columbus, United States. And Dr. Khalid Mumtaz is my mentor in gastroenterology as well. So he's very dynamic person. So Dr. Khalid.
Hello, Assalamu alaikum. My name is Dr. Khalid Mumtaz. I am really thankful to Pakistan Society of Hepatology for giving me opportunity to talk on their annual meeting. The topic that I'm going to discuss today is guidance for the statistical analysis in research. Just to briefly introduce you, I'm working as Associate Professor at Ohio State University, and I'm also Director of Research in the section of Hepatology. Before I start this presentation, I would like to share a memorable moment. This was in 2001, I believe, first or the second Pakistan Society of Hepatology annual conference. And I was working at that time, a second or third year gastroenterology fellow at Aga Khan University Hospital, and I have opportunity to present my research work at the first PSH meeting. The topic I still remember was predictors of hyperkalemia in patients with cirrhosis who are on diuretics. And my presentation was awarded for the best oral presentation. I'm still honored that I received the award from Dr. Tahir Hussain. I'm sure a lot of you know about Dr. Tahir Hussain. Today, my outline of the presentation would include components of the research, definition of the stats, basics of the statistical analysis, types of variables, outcomes, types, univariate analysis, multivariate analysis, and then I'm going to statistical scientist, which means that I have a statistician whom I guide when I am starting our research, and then my statistician give me results. I make sense out of that. I know what is going in the hindsight scene of the statistics because I have a master's in clinical epidemiology. So I know the complexity of the behind the scenes statistics, and then based upon the initial result, I ask my statistician to give me more results. Finally, I write down the results in manuscript and get it published. Let's talk about the steps to the research, the most important in the next slide. The next step, after you know that the idea is worth exploring, is to find out what database do you have to select if you already have some existing databases, or otherwise you have to find out what data variable you are going to select for this research idea. And then you have to formulate a protocol, get it approved by the IRB, ask your statistician to do the statistics. Finally, once you get the results, formulate an abstract, write down the manuscript, and get it published. Out of this first research, you can actually go ahead and do some serial research and get some grants as well. Where does the idea generate? The idea generated multiple areas in your poster area and see the posters, and then you can actually get some idea from the posters. You go to the ACG, ASLD, DDW meeting. From there, go to the poster section, read the abstract book, and then find out the ideas of what others are doing. Discuss with the pharmaceutical industry about your research idea and find some help from them or assistance from them. In my practice, during my inpatient service week, I discuss about the patients with my residents and fellows, and their questions actually are sometimes intriguing for me to go and look up the answers to their questions, and then I find that there is not much research in that area. One of the recent example is my research paper on the incidence rate and outcomes of C. diff infection in patients with cirrhosis of liver. So I see a few patients with C. diff infection and underlying cirrhosis. I looked up the literature. I found multiple reports of single center, few case reports, case series, but I didn't find any national data on outcomes of C. diff infection in patients with cirrhosis. So I looked up the nationwide inpatient sample database, and then I formulated an abstract and finally wrote a manuscript and got it published. You can discuss the research idea with your colleagues. They can actually further improve on your ideas. You can discuss with the other specialists. Like recently, in our post-liver transplant patients, I saw post-stress cardiomyopathy, which is also called as a Tekatsubo's cardiomyopathy. I looked up the literature. There's not much. I looked up at nationwide inpatient sample database, and then completed an abstract, which was presented in ACG meeting a couple of years ago, and now I'm working on the manuscript about what is the incidence and outcomes of Tekatsubo cardiomyopathy in patients who are undergoing liver transplantation. We found that about 1% population gets it, and majority of the patients are female who gets post-stress cardiomyopathy. And then you have to find out a mentor who can guide you in the field of your interest. And then you can read actually the latest journal and published studies. That's where the idea hatches. These are the different search engines that I used for searching the literature: PubMed, DW On Demand, Consensus Abstracts, and Google Scholar. After formulating, or while you're formulating a protocol, a question that would come in your mind is where to get the data from. I'm sure that majority of you are working in a hospital setting, so you can go and get the data from the hospital information warehouse. A lot of hospitals are using electronic medical record. If not, then you have to go and fetch the data manually, that I used to do some 20 years ago from the files of the patient. Here at Ohio State University, we have access to multiple large databases like PICORNet, NIS, NRD, and Truven Market Scan, which has a national database on various liver diseases and multiple other diseases. It has records of thousands of patients. Where to collect the data? In old days, as I mentioned, that we used to collect the data on the paper data collection form. Is still you can collect it, but immediately after that you have to convert it, transfer it into an electronic file, such as an Excel file, which is unstable, but a lot of people have access to that. Or you can actually transfer it immediately to SPSS file. Here at Ohio State University, we have RedCap database, which is very stable. It is a secure system of storing the data on computers. And there are a lot of centers in US, Europe, and Asia where they use RedCap. And therefore, if you're doing a multi-center study, RedCap database is very, very helpful. It's very important that you involve a statistician right from the beginning when you're writing a protocol. Here at Ohio State University, every first Wednesday of the month, we have a research meeting where we present our protocol in the presence of our statistician, Alice and Kyle, get the input from them, and then they get us the statistical analysis. You can be your own statistician. You can use different statistical software, such as for the basic research, SPSS and R are very useful, and they are very user friendly as well. You can learn SPSS by watching the YouTube videos. For the advanced research analysis, you need some advanced software such as SAS and Stata. And for operation of these two, you need to have skill sets, or you need a skilled statistician. Let's talk about the definition of statistics. Statistical analysis is the organization and analysis of quantitative or qualitative data using statistical procedures, including both descriptive and inferential statistics. Why do you need statistics? You need statistics and sophisticated statistics to add evidence to your research question. And talking about evidence, there are different levels of evidence, and it depends upon what is the quality of your statistics. You can add to the existing evidence-based medicine a 1A level of evidence in the form of systematic review and meta-analysis of randomized control trial, followed by that randomized control trial, and then there are different other grades of level of evidence in the form of cohort studies, case control studies, analytical studies. The poorest evidence in the field of the medicine is expert committee report or opinion. So if you don't have any of the upper level evidence, sometimes this evidence is what is existing, and you have to rely on that. When you talk about the different type of studies, the type of the studies start with who has assigned the exposure. Has the investigator assigned exposure? If not, then this is called as an observational study. If in an observational study there is no comparison arm, then you call it as descriptive study. The most common descriptive study is a case series, and if there is a comparison arm, then it's called as an analytical study. Within the analytical studies, there are different types of studies. Cohort study, where you have an exposure, but you look for the outcome, such as if you have patients with hepatitis B and you are prospectively following them, marching forward to find out what is the outcome, how many have developed cirrhosis of liver, how many have developed decompensation, how many have developed outcome in the form of HCC. Case control studies are the one where you have an outcome, but you don't know what is the exposure. Like in 1970s, there was a famous study about the lung cancer etiology. And when hundreds of patients with the lung cancer were interviewed and their data was looked into, a common thing found among majority of the lung cancer patient was their history of smoking. And from there, we understand and learn that smoking is one of the main factor leading to the lung cancer. Another study in the late 80s and 90s was when HIV infection came into the picture. So people were not aware of that, what is the etiology or what is the risk of HIV. And then the patients with HIV were interviewed, and one of the most common risk of homosexuality was found in those days. And then cross-sectional study, which has exposure and outcome at the same time. Usually they are retrospective cohort studies. Let's talk about the basics of the statistical analysis. In the next few slides, I'm going to talk about the review of variable types, descriptive statistics, measure of central tendency, measure of dispersion, inferential statistics, including hypothesis testing, common tests that we use, sensitivity, specificity. So in statistics, the two most common type of variables are the independent variable and dependent variable. In a data set, there are hundreds of independent variables, and then there are 3 or 4 dependent variables, which are usually the outcome variables. So when you're learning about the hepatitis B causing liver cancer, hepatitis B is a independent variable and liver cancer is a dependent variable. On the top of that, the other independent variable in this study could be a concomitant hepatitis C infection, a concomitant hepatitis delta infection, concomitant use of alcohol, leading to the dependent variable or the outcome of liver cancer. Within the independent and dependent variable, they can be categorical or numerical variable. Categorical variables are qualitative variable, which are divisible further into nominal and ordinal variable. The nominal variables include gender, race, color of eyes. They usually do not have natural order or ranking. The ordinal variable, as the name suggests, have usually orders or categories, like categories of age 20 to 25, 26 to 30, 31 to 35, 36 to 40, and onward. And then the pain scales are also ordinal categorical variable. It can vary from 0 to 10. However, as I've mentioned, that there is no natural order. So pain from 0 to 5 is not equivalent to pain from 5 to 10. So therefore, they do not have any natural order. And then the other type of variable that we come across is the numerical variable, which is a quantitative variable. Within the numerical variable, we have discrete variable. Typically, it includes the count, like if you are asking your patient how many times in an year they have developed flu. So this is a discrete variable. And then the continuous variable include weight, height, and lab values such as hemoglobin, creatinine, bilirubin, INR. There is usually a set distance between the data point. Now let's talk about the measure of the central tendency and the measure of dispersion. Measure of central tendency provide the information on the typical or average value of the data set or the middle value of the data set. There are three ways to report the measure of central tendency: mean, which is used for the continuous data, and it is usually reported with the standard deviation. On this blackboard, you can see that there are 7 variables, and if you sum up these seven variables and then divide them by 7, this is going to give you a mean. Mode is the most commonly occurring value in this blackboard. 7 is the mode, and median is the middle value of the data set, which is used for the continuous or ordinal data. Below that, you can see two graphs, the two data sets. This is data set A and this one is data set B. You can see that in data set A, the mean, median, and mode are in the middle. So this is quite equally distributed or normally distributed data set. However, the data set B is very skewed and it is not normally distributed. You can see that the mean, median, and the mode are far apart. The other descriptive stats is the measure of the dispersion. Within the measure of dispersion, there are two entities: variance and the standard deviation. Variance is the average differential of all data points from sample means squared. The standard deviation is the square root of the variance. Greater the standard deviation, more spread out the data set is. On this graph, again, you can see that if you have a value with mean with the 1 standard deviation, it means that 68% of the sample of data fall within 1 standard deviation. If your standard deviation is plus minus 2, it means that 95% of the sample fall within 2 standard deviation. And as I mentioned earlier, greater the standard deviation, the more spread out the data is and less reliable your results could be. Next, moving to the hypothesis testing. When you're formulating your studies, you must be aware of the hypothesis. There is a null hypothesis, which means that there is no difference between the groups, and then alternate hypothesis, which tells you that there is difference between the group. Hypothesis is tested with the inferential stats that gives you a p-value. Let's talk about p-value. It is the probability that the observation is caused by a random chance. Null hypothesis means that there is no difference in the development of HCC in patients with and without hepatitis B virus infection, and the alternate hypothesis for this hepatitis B statement would be that hepatitis B virus patients are at risk of development of HCC. If you have a data and you assess your hypothesis, and if the p-value is 0.05, you will be able to say that there is a 5% probability that this observed difference is due to random error and not true. However, 95% chances that this is a true statement. Although in the world of the stats, you just limit yourself to the fact that 5% probability that this observation is by random chance. When you start the analysis, there are two types of analysis that you perform: the parametric analysis and the non-parametric analysis. There are certain requirement for both these analysis. Parametrical analysis is most commonly used type of statistical analysis. It is used for the numerical data, such as quantitative data that are normally distributed. There are two assumptions: the assumption of the normality, which specifies that the mean of the sample group are normally distributed, and the assumption of the equal variance, which specifies that the variance of the sample and their corresponding population are equal. Non-parametric tests are less powerful tests. They are used to analyze ordinal and categorical data. They usually do not meet the first two assumptions that I have just discussed with you. If the distribution of the sample is skewed towards one side, or the distribution is unknown due to the small sample size, in those situations, non-parametric statistical techniques are used. Let's move on to the inferential statistics, where we compare the groups. The most common inferential statistics that we use are student t-test and the chi-square test. It depends what type of variable do you have. If you if you are comparing to the means of two groups, then you usually use student t-test. If you are comparing the frequency of the two groups, then you usually use chi-square test. This is a table that I've formulated for you to find out how to proceed with the univariate analysis. If you have two group comparison and you're comparing the means, then you have to assess if it's a parametric or non-parametric type of data set based upon the distribution. You use either independent two-sample test or Mann-Whitney test. If you're comparing percentages, then you use the chi-square test. If you're comparing more than two groups, then you can use ANOVA or Kruskal-Wallis test, depending upon the parametric or the non-parametric analysis. Let me give you a couple of examples of the student t-test and the chi-square test. In this slide, I'm giving an example of a researcher who is trying to learn about the role of low-fat diet intervention, let's say in patients with the non-alcoholic fatty liver disease. There are two groups of patients, group OA and group B. There are 10 patients in both the group. The mean weight in group A patient is 71.9 kilogram, while in group B, it is 90.7 kilogram. When I applied the student t-test, then I found that the mean difference between the two groups is about 18, and the p-value, which is hidden up here, is 0.06. It means that there is a 0.6% chance that this difference could be by a random error and not real. Now let me give you an example of the chi-square test. This researcher is trying to pose a question that is there a difference in anti-clotting drug prescribed between the two hospitals. So in hospital A, there are 15 patients. Out of these, 11 are on warfarin. In hospital B, there are 15 patients. Out of those, majority, that is 11 patients, are on clopidogrel. So apparently, it seems that there's a difference between the prescription of clopidogrel and warfarin. However, you have to prove it statistically, and when the statistical test, chi-square test, is applied, then the p-value was found to be 0.05, which means that there is a 0.5% chance that this result could be by a random error. Now let's move on. We are seeing a lot of patients with the COVID, and there are a lot of diagnostic tests which are developed for the diagnosis of COVID. And in setting of finding out the accuracy of the test, we usually use the sensitivity and the specificity. So let me talk about that. Sensitivity is a measure of the ability of the screening test to identify the positives, and specificity is a measure of the ability of the screening test to identify negatives. In simple word, if you have 100 patients who have symptoms of COVID, you have a testing kit, you go ahead and test them, and all 100 of them are turning out to be positive. It means that this test is 100% sensitive in diagnosing COVID. On the other hand, if you are checking the same diagnostic kit on the healthy controls, You check it on 100 patients, and all 100 of them are found to be negative for COVID, and they are negative. So it means that this is 100% specific as well. However, in the real world, there isn't any test which is 100% sensitive and 100% specific. Related to the specificity and sensitivity, there are some other terminology which you must be aware of. The true positive, the number of the observations the model predicted were positive, that were actually positive. The true negative, the number of the observation that the test predicted were negative, that were actually negative. And then there is false positive, the number of the observation the test predicted were positive, that were actually negative. And then the false negative, the number of the observation the model predicted negative, and they were actually positive. So far, we have talked about the univariate analysis, and in the last slide, I talked about the sensitivity analysis. In order to do further sophisticated analysis, you have to move toward the multivariate analysis. Multivariate analysis is able to tell you about a variable independently predicting the outcome or not. Multivariate analysis allows simultaneous study of multiple variables for the outcome. It accounts for the interdependence on the variable, more informative than the univariate analysis. However, it is more complex but gives more precision in the results and conclusion. There are different types of multivariate analysis. It depends upon what type of outcome do you have. It could be linear regression when your outcome is continuous, such as length of a stay or total cost of the care that you're looking for. It can be logistic regression when your outcome is nominal, such as mortality, survival, graft loss, yes, no answer. And then the Cox proportional regression is usually used for the time bound outcome or time to an event analysis, such as if you are trying to study the time to the death or the time to the graft loss in a patient with the liver transplantation, then you usually use Cox proportional regression analysis. Let me give you an example of multiple logistic regression analysis that we have used in our recently published paper. In this paper, what we have done, we have tried to find out the effect of bariatric surgery in patients with cirrhosis. In the previous studies, a majority of the literature is suggesting against bariatric surgery in patients with cirrhosis. So we have tried to study that, and on a national readmission database, our outcome in this study was mortality, inpatient mortality. So we have performed a multiple regression analysis, and we found in the beginning that cirrhosis is associated with increased mortality, as you can see here with the p-value of less than 0.01. However, we splitted the patient with the cirrhosis into compensated and decompensated cirrhosis, and we found that compensated cirrhosis as compared to no cirrhotic population do not have significant mortality. However, the decompensated cirrhosis is the one which is contributing to increased mortality. There's 83 times increased mortality in the decompensated cirrhotic patients as compared to the non-cirrhotic population. We also found that decompensated cirrhotic patients who are undergoing Roux-en-Y gastric bypass as compared to the vertical sleeve gastrectomy have 3.9 times increased chances of dying. And we also found that if the procedure is performed in low volume centers, that is where there are less than 50 gastric bypasses performed per year, there is a 5 times increased risk of death in that population. So the message is you can go ahead and plan a bariatric surgery in a compensated cirrhotic obese patients for helping with their obesity, and you can try to avoid Roux-en-Y gastric bypass, and you must refer your patient to a center with extreme experience in this surgery. So let me summarize what we talked about. We talked about the steps towards the research. We talked about the role of the statistics. We talked about the frequency table, measure of the central tendency with the help of mean, median, and mode. We talked about the measure of dispersion in the form of variance and standard deviation. We talked about the univariate analysis hypothesis. I gave you an example of multivariate analysis. Once you are able to do all these analysis, the next step is to write up your paper and get it published. But the story doesn't end here. Once you complete your publication, you have to send it for, once you complete your manuscript, you have to send it for publication to a journal, and the journal will send it to reviewers to find out if your study is really publishable or not. There are different types of reviewer. There are reviewer who would just give you some trivial comments, and then there may be a reviewer who is ready to eat up your manuscript and give very strict comments which may or may not be answerable. The message is to be persevere and continue to try. If you fail to publish it in one journal, you can go ahead and try it in some other journals. I would like to end here, and thanks again, Pakistan Society of Hepatology, and I would like to thank Dr. Zishan and Dr. Alam for this arranging this workshop and giving me an opportunity. And I wish the best to Pakistan Society of Hepatology with the rest of the meeting. Thank you.
Thank you, Dr. Khalid Mumtaz. Wonderful talk, and I love the cartoon regarding this publications issues. So the next topic of presentation is very important: how to get your manuscript accepted for publication. And I would like to invite again Professor Adil Ajwad Bhatt for the presentation. Dr. Adil.
Once again, I would like to thank everyone at PSH for allowing me to present this, particularly Dr. Altaf Alam, Dr. Zishan, and all the other key members who have made this happen. So this is the second part of my talk, which is how to get a paper published. Earlier on, we talked about how to start a research project, and this is assuming that now you have completed your research project and your paper is ready, or you're about to start writing the paper. How do you get this published in a reasonably good journal? And once again, the caveat is that I will focus on clinical papers only and will not talk much about bench research, bench papers, or lab papers at this time. Here is a broad outline of my next 20 minutes of talking. We will talk about attributes of a good scientific paper, a brief discussion about the difference between abstracts versus a full-length paper, the components of a scientific paper, things you need to do before you start, a few general guidelines followed by some specific points, anticipating critique, and then what would be the next steps after you've done all that. So here are some of the attributes of a good scientific paper, a good clinical scientific paper. It, at the very outset or at the very minimum, answers a novel or an exciting question. A good scientific paper is rigorously designed and executed. In general, randomized clinical trials receive the highest priority in most journals, but other designs, if they are well executed and well thought out, including appropriate populations and do the correct analysis, also have a high chance of acceptance. A good scientific paper has to be very well written. In clear and simple language, there should be no spelling, grammar, or syntax mistakes. It should follow a logical and understandable sequence of events. It adheres to ethical and regulatory guidelines, which include IRB approvals, patient consents, and listing in trial databases before you have started your research if it is a clinical trial. What is the difference between an abstract and full-length paper? We often write abstracts and present at professional society meetings. Now, abstracts generally present only preliminary results. They highlight some of the key findings of what you've done, but not the details, because you may not have the details at that point in time. The methodology that you describe is relatively concise, and the results are that you present are rather focused. There is little less rigor in writing an abstract and in peer review of abstracts compared with a full-length paper. In contrast, a full-length paper that you submit to a reputable journal addresses or presents your final results of the project or your study. You as an author or the senior or corresponding or the lead author must be able to certify the authenticity of these final results. You need to provide detailed methodology, your full results, any additional sensitivity analysis if they're warranted for the reviewers as well as the readers to understand exactly what you did and the whole context of this. The full papers get much more rigorous peer review, both in terms of content and pre-publication scrutiny. A few things before you submit is you need to choose the journal you're submitting to wisely. And a lot of factors will go into how you choose a journal. You need to think about the relevance of that journal to your research. For example, there is no point sending a hepatitis C or a NAFLD article to an obstetrics and gynecology journal or a cardiology journal. You need to figure out a general medicine journal or a hepatology journal or a gastroenterology journal for such topics. Consider how rapidly they review your paper and get back to you. And these data are often provided on the journal websites. You need to understand who the audience of that journal is. Who reads that journal? Are those the people that you want your paper to be read by? Then you need to look at the impact of the journal. Generally, you want to publish in a higher impact journal, which is indexed and reaches a lot of people, because academic progress and academic appointments often depend on the impact of your work. Also consider if the journal is an open access versus a non-open access journal. Try to avoid what we call predatory journals who just take money and would publish anything that you submit to them without any good rigor. And there's a lot of those journals these days who would be writing to you every single day. Now, every journal has different requirements for acceptance and submission. So it is very important that you read the instructions to author very, very carefully and follow them diligently. Follow the font, the margins, the line spacing guidelines, and assemble all your materials before you start uploading the files on the journal website. And we will talk about what are the materials that you need to in order to submit. In certain cases, you may be able to write to the editor or a deputy editor before the submission to gauge their interest and if your paper is among priority areas for that journal. All that requires is finding the email of the editor from the website and dropping them a short email or a letter saying that this is what we have done, this is the topic of our paper. Is there something that you or your journal would be interested in? And no editor will commit to you that they will publish it. All they will say is we are interested or in reviewing it, or this is not something that we generally consider. It's the latter that helps you more and saves you time. If they say outrightly that this is not an area of interest for us or for our audience, it saves you time of submission and waiting for them to respond to you and then submitting to another journal. Again, before you start the study, if it's a clinical trial, you have to get it registered at a publicly available website or a registry like clinicaltrials.gov. Most reputable journals will not publish or even accept or review your paper, clinical trial results, if they have if your trial has not been registered at these sites. You have to follow the published guidelines, and these guidelines are published by ICMJE, International Council of Medical Journal Editors. And one example of these guidelines is the Consort Guidelines for Clinical Trials. Other types of studies like meta-analyses, cohort studies, etc., have their own guidelines which you need to follow. And get all your ethical board, IRB, all regulatory approvals, and you have to put them in your paper before you submit that. This is just an example of randomized clinical trial consort guidelines. This is how journals want you to present how you created your data set or your study groups. And these are, again, available on journal websites, so you can take that. And it also is helpful to look at that even before you design your study, because this can help you understand what are the steps that you need to undertake to design a rigorous trial. Before you're ready to submit, you need to gather some basic information about everyone who participated in writing that paper and other important aspects of your paper. You'll need the full names, email addresses, institutional affiliations for all authors. So have that ready when you're ready to submit. Most journals these days ask you for names and contact information for potential peer reviewers. They may ask them to review or they may not ask them to review, but they generally make you put that information in there. So have a list of reviewers ready on your computers. You can just plug that in when you're submitting a paper. A cover letter is required by most journals, and this cover letter should include the title of the paper that you are submitting, but also a paragraph about why your research is important to the audience or the readership of that particular journal that you are submitting to. Tables and figures need to be uploaded as well. Some journals will allow you to have tables and figures as a part of your full manuscript as one file. Other journals ask them to be uploaded separately as an individual files. For example, figures may be required to be uploaded as a JPEG or a TIFF format or a PNG format. So read the instructions to authors carefully because different journals have different requirements for that. Let's move over to what are the components of a scientific paper and how do you write that so that it is reviewed favorably by a good journal. This is generally the order of submission of these things. You have to have a title page, an abstract, introduction, methods, results, discussion, your references or bibliography, and any supplementary material. And we'll go over that in detail one at a time. So title page is the one where you put the main title of your paper. Generally, we suggest that it should be a catchy title that piques the interest of those who are reading and those who are reviewing, and it should reflect a major message from your study. Some journals have word limits on this, so be aware of that as well. This title page has to contain all author information and their affiliations. You must have keywords in there. Generally 3 to 5 keywords are required for most submissions. This helps the journal to index once they have accepted the paper. Many will ask for a word count, the number of tables, figures, number of references, etc. Some ask even for the number of pages that are in the manuscript. And you have to have all funding and disclosures, acknowledgements on this title page as well. You also need to put an authorship statement which details as to what was the contribution of each of the authors that you've listed out there in terms of writing this paper. And on the right side of the screen is an example of a title page of a paper that we submitted about a year or two ago and was published in a highly reputable journal in infectious diseases. And it has all the information that I talked about, and it has a second page as well, which I am not showing out here. Next part is the abstract, and some journals ask for, most journals ask for a structured abstract, although some may be okay with a non-structured abstract, and I'll show you in the next screen what that means. Now for abstract, word count is critical. You have to follow this word count of that particular journal very carefully because they will not allow you to submit the paper even if the word count is 1 word over the limit that you're asking. Make sure that your most important result and message is in your abstract. You have to have just enough information on methods and study population, but the most critical part of the abstract is the most important message that you want to convey and the strongest results that you have. Remember, most people will only read your abstract. Hardly a very small proportion of people will go on to read your full paper. So whatever information you want to convey to everyone should be contained in this abstract. On your left is an example of unstructured abstract. On your right is an example of a highly structured abstract. So for example, till a couple of years ago, Hepatology, one of the leading journals in liver diseases, allowed us to submit papers with the unstructured abstract. So this is just a free-flowing narrative that you put together without any subheadings. Another journal, for example, JAMA, a group of journals, requires a very, very detailed, highly structured abstracts, including subheadings like importance, objectives, design, setting, participants, main outcomes and measures, results, conclusions and relevance. And they often ask us to put limitations in there as well. So this is the example of a structured abstract. Again, you have to read the journal's instruction to authors to make sure that you conform to that. Now you come to the main part of the paper. The first part is introduction. This is about 1 to 2 paragraphs where you put the background and significance and any gaps in knowledge in the field that you are studying. This is not meant to be a comprehensive review of literature. It is only there to set a stage for what you did in the study and to provide your specific aims and or hypotheses that you tested in this paper. So all you need is about 1 to 2 paragraphs of setting the stage for people to understand what you did and why you did that, and what are the gaps that you're trying to fill through your paper. Next part, which is a critical part, is the methods part. In this section, you may again have either an unstructured free-flowing narrative What I generally do for full-length papers is I divide that into subheadings and make it structured so that I don't miss anything, and it is easier for the reviewers as well as the readers to understand what I did and how I did it. So what I do is I subdivide it into design and settings and participants. So this tells about what is the kind of study design I used, where did I conduct the studies, and who did I include in my study as study participants. Then I talk about data sources. Where did I collect the data from? If there were any interventions, what were they, and what were the primary outcome measures, and how did I measure that? If there is any randomization or blinding in a clinical trial that you need to put into this section out here. If you matched the groups, for example, propensity score matching or inverse probability of censoring and treatment weights, you need to give details of how you did that matching of the groups. And then you go into statistical methods as to what kind of statistics you use, and Dr. Khalid Mumtaz gave you a very good overview of how to do this part. This also includes your power calculation beforehand as to how you arrived at the sample size that you are presenting. You have also to address what did you do with missing data, how much missing data was in, how did you address that. And then you come to the results part. Here, again, you divide it into paragraphs. The first paragraph generally is about baseline characteristics of your study population. You can give descriptive data out here, some outcomes data in the next paragraph. And then you go on to next paragraphs where you provide other main results, like, for example, your primary outcomes and your secondary outcomes. You can structure them into your unadjusted and adjusted results, and then provide any subgroup analysis or sensitivity analysis. Again, as Dr. Khalid Mumtaz pointed out, you have to give significance levels, dispersion metrics like confidence intervals, etc. I suggest don't be too heavy on text and numbers in this part. Give main results. The rest can be presented in your tables and figures, and you can just refer them to that in your text that for further results, look at the tables and figures. Now, how do you structure tables and figures? There is generally an unwritten rule, which is mostly accepted by, by authors and journals. Table 1 generally provides baseline characteristics of your study population. This is before the outcomes have occurred. So you provide the demographics, you provide the clinical characteristics, any lab values that you need to present, and so forth. And you do comparison of your populations if you have more than one study group. Your table 2 is your main comparison of characteristics of the population versus controls, or your main outcome result. And table 3 onwards then gives more information on different types of modeling and more detailed statistics, or your secondary outcomes that you are presenting. So just follow this basic outline of presenting tables. Figures, generally figure 1, if you have figures, if you have created a study flow, I always use figure 1 as my study flow sheet or cohort construction. In clinical trials, it's the consort study guide, the study table that I showed you earlier on. And then figure 2 onwards include comparisons of your groups, your main results if they're presented in a graphical manner. You can put Kaplan-Meier curves for time to event analysis, for example, bar charts, histograms, forest plots, scatter plots, etc. Depends on exactly what you are presenting would determine what kind of figures go into this section. Then you come to the discussion part. And again, this is divided into several paragraphs. Generally, what most of us do is first paragraph is a very short paragraph which describes or summates your main result in 2 to 3 sentences. We do not go into the details in this particular sentence because some people will only read the first paragraph to understand what your key point was. Paragraph 2 onwards is where you provide context to your results that you found. How do you interpret those results? You compare them with what is previously known and published, and try to give some reason of why you found what you found. And you can have 2, 3, 4 paragraphs here after the first paragraph, depending on the number of things that you want to expound on. But generally, the structure of these paragraphs should be, as I said, they should provide the context of your findings in view of what is largely known to the world, and how does your study differ or conform to existing literature, and what are the explanations of that. Then you come to the second last paragraph. Here you put in limitations and strengths of your study. uh, what were the biases that may have affected your results? How could you have mitigated? How could you have mitigated these results? Are they generalizable? What are the strengths associated with your study that people should consider? And then you come to the last paragraph, which is basically your conclusions and what is still unknown and what people should focus on in terms of future studies. In other words, this goes on to what did you find? What is the explanation of your findings? What did you miss or could have done better? Why is this finding important? And what do you need to do next? What happens after you've submitted a paper? So once you've submitted, one of four things will happen. The most common thing for big journals is rejection without further review. So you'll get an email within 2 to 3 days or maybe a week or so that we decided not to send this for the peer review. Very, very rarely a journal may accept your paper without any further review. I've had this once out of 178 papers that there was no further comment from either the editor or the reviewers, and they accepted the paper. Occasionally or frequently, they will ask for editorial clarifications, or they want to modify your study. You missed a key part of their submission requirements, so they'll send it back to you. And in a small proportion of cases, they will send it out for peer review. So most high-impact journals send less than 5% of the submitted papers for peer review. So don't feel depressed or dejected if they have rejected your paper on the first go. What if you get rejected? If you get rejected without review, you can submit to another journal without any changes, which is what I mostly recommend, and I do myself as well. If it was rejected after peer review, you can submit to another journal with or without addressing the comments from the reviewers. You'll need to read the comments and see if there were reasonable comments or unreasonable comments. Can you make those changes or can you not make those changes? Would making those changes make your paper stronger or not? If they ask you to revise and resubmit, make sure that you answer every single point, comment, or critique that has been provided both by the editors and each of the reviewers. You actually need to copy and paste each comment in your cover letter and then answer that saying that this is my response to you. Even if you do not agree or do not do that, you still have to respond to that saying why you did not agree to that. Be very deferential to reviewers and respectful to them even when you disagree. It may be a very nasty comment, it may be a very unreasonable comment, but you have to be very respectful and careful when you are responding to that. If reviewers ask you for new things and you're unable or unwilling to do them, explain why you cannot or will not do that. I've had many papers where reviewers have suggested to take a totally different direction and answer questions which were not at all part of my objective. And I very respectfully tell them why I shouldn't or wouldn't be doing that. As I said, most high-impact journals accept less than 5% of submitted papers, so don't be discouraged. But to make it more appealing to the journal, ensure that the language, spellings, grammar are flawless. Reviewers get very upset when they find these mistakes because they consider these as evidence of poor scholarship or lack of attention to detail and sloppiness of your work. You may need to submit to several journals before a positive response, and sometimes up to 6 or 8 or 10 journals. To give you an example, my last 10 published papers, which were all in this year, I required to submit them 31 times. So one paper was accepted, the first journal I submitted to. One paper required submitting 5 journals. But I start with the very high impact journals, which have a very high rate of rejection. So I need to submit sometimes up to 3, 4, 5 times. The highest number I've had to submit to is about 10 times before acceptance of a paper. So summarizing, here's how you need to strategize after the paper is written. Choose the journal and choose the journal wisely. Format it carefully according to their requirements. Assemble all required materials that I mentioned in the previous slide, and then you hit the submit button. Repeat these steps 1 to 4 as often as needed. In summary, a good paper requires a lot of work, A lot of thoughtfulness goes into putting it together. A lot of collaborative effort is needed for that. Choose novel questions, choose rigorous methodology, and pay great attention to detail when you're writing that. Write in simple language, concise language, in an orderly manner. As I pointed out in previous slides, it should follow logical sequences. Make sure all ethical and regulatory requirements have been adhered to, and persistence pays off. I will stop here and happy to take any questions as per the moderator's.
Thank you, Dr. Adil Ajwat Bhatt. Very nice presentation, and I have learned a lot from your presentation. Inshallah, after this presentation, I will able to publish my research in NEJM. The next presentation,
Inshallah.
inshallah. So the next presentation is how to critically appraise a paper. And for the presentation, I would like to invite Professor Arshad Kamal Bhatt from Lahore. And I also want to mention one thing, that this session, research workshop session, is mashallah overwhelmed with the participation of Bhatts from Pakistan. Dr. Arshad Kamal.
Bismillahirrahmanirrahim. Ladies and gentlemen, Assalamu alaikum. Good morning and a very warm welcome to all the distinguished colleagues who are attending this not to be missed 20th Annual Congress of Pakistan Society of Hepatology. I would like to express my gratitude to the organizer of this conference for giving me the honor and the privilege to be part of this academic event. I must also appreciate the decision by the chairman of scientific committee for devoting one full session to research and research-related issues, which perhaps did not attract much attention in the years gone by. This is a step in the right direction, and I'm confident that this will go a long way in promoting research in Pakistan. Now, I've been asked to talk about how to critically appraise a research article. And in the next 20 minutes or so, I will talk about this topic. Okay. I have no financial disclosures to declare related to this presentation. Now, the topic of critical appraisal is a huge topic, and it's not humanly possible for me to cover all the aspects. And therefore, in the interest of time, I will be focusing on specific parts of this topic, specific issues of this topic. I will start with defining what is critical appraisal, and then go on to why do we need critical appraisal? Why should we do it? What do we hope to gain from it? And then a few words about how to critically appraise a paper. We'll go through that exercise. And finally, we will look at some of the tools which can help us in improving or streamlining the appraisal process. In other words, help us in improving our skills to be better appraisers. What will not be discussed is how to retrieve articles from various resources. I'm sure the audience are fully conversant with the techniques. We have lots of databases available, PubMed, Medline, UpToDate, and so on and so forth, and we all know how to obtain articles of interest from these resources. I will not talk about how to perform statistical analysis. As researchers, we only need to know which test to use, when to use, what are the requirements of the test, and how to interpret the results. That's it. The mechanics, the mechanisms, and the mathematical background of a statistical test are not our concern. That's for the statisticians. And finally, if we are reviewing an article as a reviewer, we are expected to write a constructive critique at the end. That is not the topic of today's discussion either. Now, when we approach the issue of critical appraisal, we need to begin by answering two questions. One, what is critical appraisal? Two, why do we need to do it? The simplest definition of critical appraisal is that it is a process which systematically allows us to examine research evidence obtained from various resources. And by doing that, we are in a position to evaluate the trustworthiness of this evidence, whether it is valid or not, and how relevant it is to a particular context. That is the definition of critical appraisal. Now, please note, that the word systematically appears in this definition, which means that it is a systematic process. It's not a haphazard process. It has to follow a certain pattern. The second question was, why do we need to do critical appraisal? What's the use? To answer this question, I will take you back to the early 90s and give you a glimpse of how the practice of medicine has changed. In the pre-90s era, whenever there was a problem, a critical issue, we used to sort evidence, we used to sort advice from experts, from our seniors. In teaching institutions, the word of the professor was the final authority. It was an unchallenged opinion given by the professor, and there was no way anybody could disagree. We all followed that. It all changed in 1991 when McMaster University introduced the concept of what we now know as evidence-based medicine. And this has gone from 1990s to the present day as well. The evidence-based medicine allows us to ask for the reason why this, ask us questions, why we want to do something, what is the evidence of its utility. No longer it is enough to say it's my experience, but it's my advice. I have to come up with evidence, convincing evidence. So what is evidence-based medicine? It's the process of making clinical decisions about a patient's condition or clinical state, which takes into account the best available evidence, no matter how we have obtained it, coupled with our research expertise and patient preferences, informed decision by the patient about management. This is evidence-based medicine. And there are 5 steps to it. We first ask a question, then we collect evidence, then we critically appraise that evidence, and when we see that we can apply it or not, and finally we monitor whether the application has produced the desired results or not. And you will notice that critical appraisal skills sit right at the center of this 5-step process. If we are not comfortable with our critical appraisal skills, if we cannot interpret the information provided in a journal article, then it becomes an exercise in futility. We will not be able to apply it, and we will not be able to improve our patient management strategies. There has been an explosion of medical knowledge. When I was doing my residency way back in 1980s, and look at the journey from 1980s to present, at that time, very few library resources were available. I had access to just 1 or 2 books, and to get information about new articles, I had to wait for 3 months before the next printed version of Index Matrix would arrive in the library. And then there were long waiting times for getting access to full-length articles, months at a stretch, and sometimes they're not available. All that has changed drastically with the availability of internet. Now we have internet information highway, which is overloaded. Earlier on, there was dearth of information. Now it's an exact opposite. There's too much of information. And if you look at the magnitude of information explosion, it has been estimated that every year close to 40,000 research journals are published, which means that more than 2 million research articles are published every year by various journals. And if you look at our own medical fraternity, simply in primary care journals, close to 72 articles are published per year. It's humanly impossible to read all these. So we need a methodology, a mechanism, or a method by which we can separate what is insignificant, what is not relevant, what is unreliable and unsound from what is salient and crucial to our needs. So to answer the earlier question, why do we need to do a critical appraisal? The answer is very simple. We wish to keep ourselves up to date with the latest knowledge, because by doing so, hopefully this will translate into better patient management. However, there's just a small, little tiny problem with this approach of trying to keep ourselves abreast. If we want to keep ourselves up to date with the available clinical evidence, we need to read 19 research articles per day. 19. And it's quite obvious that it's not possible to do that. And that is where critical appraisal comes to our help. Now, the pathway of critically appraising a research article, basically from a conceptual point of view, takes 2 issues into consideration. First, we have selected an article, fine, according to our own interest from one of the resources, we first perform a quick review. It's sort of a filter. Is this article relevant? Is it worth the investment in time? Should we read it or not? And after that quick review, if it has passed that test, we go to a detailed review. We thrash out, dissect out that article in detail, and we've either come to the conclusion we should be accepting the recommendations of this article or we should be rejecting it. The idea is to ultimately to have improved quality of patient management. So let's look closely at what is the anatomy of a quick critical appraisal. The first step, we need to look or focus at two issues, the title and the abstract. The title should clearly and very explicitly state what this study is all about. The author or the authors must clearly state what is the focus of this study. And then we look at the abstract, we look at these 5 issues. What's the purpose of the study? What are the participants, the characteristics of the participants? How was it done, or what are the summary of a reference to the material method should be there? If there was an intervention, what was the intervention and what was the outcome? Look for these 5 items. Once you have decided that yes, this article is worth the time, then we move on to the next issue, a detailed critical appraisal of any selected article. And if you look at any selected article, most of the time, it's divided into 4 sections: the introduction, the methods, the results, and discussion and conclusion. There could be minor variations, but by and large, this is the theme. And the purpose of going through these 4 steps is that we want to evaluate the quality of the study design, and we want to see how reliable are the results presented, and are these results applicable to our own practice or not. So how do we critically appraise the introduction? First is, does the article attempt to answer the same question as your clinical question? In other words, is it relevant to us? Secondly, is it recently published, which should be within the last 5 years, or it is a seminal article, which means that although it was published a long time back, but the implications are very important and it has strongly influenced later development. Have the authors presented a clear hypothesis? What do they want? What do they expect of this study? And is this journal peer-reviewed? Please remember, peer-reviewed journals are more reliable because they have a robust system of check and balances incorporated into their system. The next is you critically appraise the material and method section. Is the study design of this paper valid for the study question or not? Have the authors very clearly mentioned the inclusion and exclusion criteria of the participants? Has there been a conscious attempt to limit bias, and various types of biases are there, and what the authors have done to identify the confounding factors or confounding variables, and have they attempted to minimize the influence of those confounding factors or not? And are the outcome measures valid for the health condition which we are researching? Are they applicable to our scenario as well or not? And then we look at the results. Have the subject demographics been explained fully? And if the study, there were patient groups, were they similar to each other or were they different? And can we generalize the subjects to our own local population as well? For example, the study was carried out in Europe or in States. We are not a clone of the European or the American population, but can we generalize to our own patient population or not? What statistical tests were used? And were they appropriate to the study question? Have the results been presented within the paper or not properly, in the form of tables preferably? And what is the statistical significance? Has that been identified properly? And then we come to the last section. How do you critically appraise the discussion and conclusions? The discussions must be focused on results. And it is not sufficient just to mention the results here. In fact, results should not be reproduced. They should be discussed in the discussion session. And if we find differences between what has been reported in the world literature and what has been found in this particular study, these differences must be highlighted, and the author or the authors must discuss them and try to justify, to present the reason why they are different from others. The conclusions must be based on data. In other words, The data used should justify the conclusions. The author or the authors must identify the limitations, the strengths and weaknesses of the study. And finally, the conflict of interest must be very clearly mentioned because, for example, if I am receiving some emoluments from a pharmaceutical industry or I am employed by the pharmaceutical industry and I am presenting research on a particular drug which is beneficial by that pharmaceutical industry, it is likely to be a biased opinion. So the conflict of interest must be very clearly identified. And finally, and most important, will the results help me to manage my patients in a better way or not? This is the conclusion which we must draw. Now, how do we go about doing all this? The appraisal methods actually depend on this type of study design. As we know, there are lots of study designs, and appraisal methods will vary slightly on depending on which particular study design we are concerned with. And in that context, we need to be cognizant of the fact that there are primary research articles, which are the expert opinions or editorials or review articles, and then there are cohort studies, case reports, and randomized control trials. These are primary research and these are not appraised. They have to be appraised by the reader. Higher in this hierarchy pyramid are what we call secondary research issues, the systematic reviews, the meta-analysis, the evidence-based guidelines published by the various societies. This is secondary research and these are pre-appraised and therefore they are far more reliable. So we need to be cognizant of the fact what is the hierarchy of research that we are evaluating. Now, how to facilitate or expedite the appraisal process? Is there a way? Yes, we can do that. Because checklists, one way of using them is to use the checklists which are freely available on the internet. My two favorite ones are the checklists of Center for Evidence-Based Medicine or CEBM and Critical Appraisal Skill Programs Test checklist of CSP. So I'll quickly go over the CSP checklist. Lots of them are available. In fact, they cater for every possible study design and they're freely available at this website. Let's talk about the CSP cohort study checklist. We can look at these checklists and employ these checklists to have a semi-objective evaluation of a research article, starting with, is it a clearly focused issue or not? Was a random selection applied? What is the accuracy and validity of measurement of exposure and measurement of outcome? Are the confounding factors identified and minimized? And was the follow-up complete or adequate or not? And then you score it accordingly. The next section of this modified list addresses the results. Are the results clearly presented? Are they precise and reliable? Can we apply these results to our local population? And do they fit with other available evidence? And are there important implications of these results for our current practice or not? So, ladies and gentlemen, in conclusion, appraisal skills are a must for practicing evidence-based medicine. We need to learn these skills if we want to be good researchers. Why? Because this helps us to exclude research which is poorly conducted, poorly designed, unreliable, and cannot be used to inform medical practice. Appraisal skills help us to combat the huge information overload which is present in the internet, and it does so by selecting and identifying what is clinically relevant and what's not. And to make this process easier and more profitable, we can use checklists. Anybody who is interested in the process of conducting an appraisal of research articles, I would strongly recommend these two reading materials. One, The Doctor's Guide to Critical Appraisal by Gosal and Gosal, published way back in 2015, an excellent book, a very short introduction, but gives you a very pragmatic and practical advice on how to approach this problem. And the second one is How to Read a Paper, the latest edition appeared last year. It's the sixth edition, written by Trisha and Greenhaig, again an excellent paper. Anybody who's seriously contemplating becoming a good appraiser should have these two monographs on his or her bookshelf. So thank you very much, ladies and gentlemen. This brings us to the end of my presentation. I would like to thank all the participants, the faculty, organizers, and sponsors. Thank you very much, PSH 2020. Thank you very much indeed.
the critical appraisal of the paper. So the topic of our next presentation is how to write letter to the editor, and it's a very important topic. And I would like to invite Dr. Nadeem Thiami, who is consultant gastroenterologist at UHS, United Kingdom.
Assalamu alaikum and good morning to the participants. It's really good to see the interest and participation level. I've really enjoyed the talk so far, and I hope this talk will nicely fit in after previous talk by Professor Butt. Could I also like to thank the organizers for the kind invitation. Now, I would like to divide this talk into three parts. We will discuss why to write a letter to the editor, and who to write, and then we'll discuss what to write in the letter to the editor. So what are these letters? So I think, I guess the research is a daunting process for most clinicians. But letter, I guess, is a very good start for those who are embarking on this journey. Letters are essentially a control mechanism that facilitates the progress after an article has been published. Before I go any further, I would like participants to look at this image on the screen and try to guess who this gentleman is. I'm sure most of you will get it right. So let's talk about what is the need? Why should we be writing letters? From an accomplished professional point of view, it's really hard to find time from busy work and family life to sit down and write a letter. Plus, letters to editors do not hold a high value in the academic world. And from a trainee's perspective, especially if they come from a setup which is not very research oriented, they may not have the skills to critically appraise a journal and then write a letter to the editor. Writing still gives you an opportunity to share your interpretation of the published data. Now, academic world, as I see it, it is very challenging from different aspects. Again, as I said, for most clinicians, we do want to write. At the same time, we do not want to hang our dirty linen in the public. But we know that letters make difference and they can influence the history. So the image on the screen is of 1860 Republican candidate for the presidency, who we all know as Mr. Abraham Lincoln. He was written to by an 11-year-old girl, stating that he had a thin, lean face and he should grow a beard, as all ladies liked whiskers. And we know that letter initially was not appreciated very much by him, but it made a difference to the history that we know. So letters helps us in developing this communication skills. And I guess communication skill is probably the most important skill that a doctor need to have, probably more important than the knowledge required to treat your patients. We would like to think that the readers are smarter than the reviewers. We know all letters, as Professor Butt had already shown you, go through a very stringent vetting process before the publication, but still some issues can go unnoticed. And in this situation, readers get an opportunity to offer their interpretation by writing a letter to the editor. The International Committee of Medical Journal Editors recommends that publication to these letters in journals should be published in the journals together with the answers. And we know there are some midline index journal who print these letters with their response under the title of the original article to prevent their escape from the attention of the readers. There are different types of the letters. So commonly we see letters written in response to the previously published articles. It could be a brief feedback on the published clinical data or the research data. There can also be a feedback on interesting case reports or case series. There are some letters written uncommonly on journals policy, quality, or format, although they are not very encouraged. But they can also be a way of accessing study material, or could be a way of sharing ideas for future collaborative study. Now, I, as a clinician, believe that my hands work better than my head, and therefore the journals I read are mostly endoscopy related journals. And the next two diagrams, including this one, the one I'm sharing with you, will hopefully highlight that there is always room of improvement. So this flow diagram that you see comes from an international consensus statement for endoscopic management of distal biliary stricture. Now it comes, as I said, a consensus, international consensus. Now, if you see on the left side of this diagram, so resectable pancreatic biliary malignancies these days in most HBB centers go straight for surgery rather than wasting time, I would say wasting time for acquiring tissue diagnosis. And if you look at this side of the diagram, so indeterminate masses or cancers which are deemed unresectable, the referring physician or surgeons would request decompression of biliary system and stenting. But we then know that if the patients are deemed suitable for any palliative chemotherapy, then they would ask for a tissue diagnosis. And once a system has been stented, then it becomes really hard for the endoscopist to obtain the tissue diagnosis. So from research point of view, this may be the right flow diagram, but as a clinician, I would personally like to make some changes. And when I shared it with other colleagues around the region, around the country, most agreed with the proposed changes. So what I'm suggesting here is that despite an international consensus, there is some room of improvement in this flow diagram. Now, likewise, this was a recent publication in ACG on the management, so guidelines on the management of chronic pancreatitis. And it was published by a very reputable group of authors in the field of pancreatology. But I hope you can appreciate that what it shows, that there's a number of hoops that one has to go through to reach a negative diagnosis. And is this really relevant and practical to our clinical practice? I have lots of doubts. And most interestingly, and what I find really hard to digest, is the recommendation of pancreatic histology. And again, I know from my experience and sharing my experience with colleagues around the region and around the country, I can say with a lot of confidence that this is not a practice that we have in our part of the world. So hopefully what I've shown you here from two very reputable peer-reviewed journals, that there are published data that can be challenged very professionally. Now, who should we write? Again, as I said in previous talk by Professor Adil, but he suggested obviously maybe choosing a journal based on their impact factor. And I think that is certainly the way forward. But you have to start from somewhere. And the best way of once you've learned how to critically appraise a journal is to join a group of like-minded professional colleagues and maybe start a journal club. And we did recently. And I know next morning you may find you're the only person attending it. However, the way this journal that I'm alluding to, participants were encouraged, because the juniors who were participating were encouraged that if there is a lot of discussion, if their presentation generates a lot of discussion, then they would be given an opportunity under a leadership of an accomplished professional to write to the editor and challenge what the data that they have already presented. It's probably best to choose a journal that is aligned to your professional and clinical interest. But at the same time, one of the other advantages of joining these journal clubs is that if you want to stay on top of the published data, hopefully there may be other readers or other participants who might have interest in reading another journal which might not address your areas of interest, yet it will add value to your clinical care. There are certain ground rules that we need to be aware of, as already alluded by previous speakers, and those are really important to know before you start writing, and that is there is going to be some limitation on the numbers of authors, words, figure, tables, references that you can use in your letter to the editor. Some journals might not accept letters to the editor addressing points raised in case reports or case series. There may be a time limit, and there may also be a number of pages that you can write your letter to editor on. This is a recent example of a letter that was sent following the journal club that I've mentioned, and it was accepted in GIE. It was so one of the other advantage I see of writing these letters to journals is that accomplished professional can support juniors in writing. So in this letter, we made the presenter as among first three authors, and their name was given, and they had the opportunity to correspond to with the editor as well. So two things to highlight from this slide. One is, what is the anatomy of a successful letter? Start off with a title intro, and in the initial you would like to acknowledge the study. Now you could see an obvious you could say an error, but given the limitation on the words, the author missed summarizing the article, and this is what the editor highlighted, that needed to be included in the letter so that reader didn't have to cross-reference the original article. This should then be followed by maximum 2 paragraphs of the real points that you would like to share with the editor, readers, and the authors of the original article. And in the end, summarize it. One of the other points you would see from this letter is that, yes, you would like to see a response by the author once you've written to the editor, but it is not a mandatory requirement. So you could maybe save some words by avoiding that. Some do's and do nots. We would really like to avoid controversies, but if you believe that has a direct impact on your patient care, then asking for clarification is always a good idea. Always think of a catchy title and make it easy for your for the readers of the journal. Follow the instructions, really important. Avoid the biased opinion and based your comments on the available scientific evidence. If you believe there isn't much evidence available, then you have to think of a way of giving it slightly different perspective. But if there is evidence available which wasn't referenced in the original article, you may want to present that. There are certain things in our culture which obviously impact on our writing, and the examples is you see a lot of emails with starting off and letter starting off with respected sir, dear sir. I think one of the things that I've learned here is that we should not be obsequious. Yes, you have to respect your seniors, the authors, professionally, but please don't be obsequious. On a professional level, you should be able to challenge anyone and everyone respectfully. Avoid just common statements such as more studies are needed. And once you've written a letter and then you think it probably doesn't add much value to the research work, then probably just think twice. Do you really want to send this letter to the journal? Now, I'm grateful to my colleague Keith, who shared this slide with me. So I think his top tips are that once you've read a journal, an article in a journal club, then, and if it has generated a lot of interest, then think, is this an opportunity for me to write to the editor? Now, remember, the more you write, it will improve your clinical skills. You will also be noticed by the editor, even if your letters are being rejected. And I've seen occasions where editors have written back to the authors after having at least 2-3 rejections and encourage them to write letters on another study that they thought might have a potential of getting accepted. And then they've also offered some collaborative projects. So if you think there is a value in writing, then plan it, go through the available literature, go through what the journal's requirements are. Once you've written it, share it with your colleagues from that journal club, preferably. Let them review it, take their feedbacks on board, make changes as required, and then try to just focus on 2 points that you really want to highlight in your letter to the editor. Make sure once you've written a letter, it has a flow, it respects the rules of the journal, and then submit it. We can still mess it up if we think the comments made are not very relevant. If we have reiterated the points which are already mentioned in the discussion sections, there are points which are very ambiguous and do not make any real change to the practice or the conclusion made by the original article. And if you think there's a lot of faff about the latter, and it doesn't really, it's not very precise or concise. Uh, no point in including lots of, I mean, so we live in a world where we've got lots of infographics around us and lots of videos and pictures around us, but in that letter it's probably best to restrict yourself to maybe one table or one figure. Language again is really, really important, because essentially, as I said earlier, communication is the key. So you may come across, I'll give you an example recently where there was I had a colleague where we were trying to really struggle to describe him in his absence, and he wasn't very tall, so I wanted to say how to really put it to the audience. And one of my colleagues came up with or the words he uses, "Oh, do you remember that gentleman who was vertically challenged?" What he was trying to say that person wasn't very tall. So what he did, although he said the same thing which I wanted to convey, but had a better word for it. So if you can't communicate with choosing the right words, professionally right words, it will have an impact on or increase your chances of getting it accepted. And really important to avoid any comments on the credential or competency of the author. What you're talking about what is being said, not who said it. Essentially, what Hazrat Ali quote was that you really want to focus what is being said, not who has said it. So in conclusion, Mr. Chairman, ladies and gentlemen, we would like to keep it short and sweet, brief and conclusive. A letter to editors should deliver an understandable message, should comply with the journal format, contain adequate and important information, and should be written to deliver objective and constructive comments. Thank you very much for listening.
Thank you, Dr. Nadeem Thiami. So I would like to invite Dr. Shahid Sarwar for the post-workshop assessment MCQs.
So you can see my slides.
Yes, Dr. Shahid, please continue.
So as we are already running late in our session, so in the interest of time, I will again go through the same multiple choice questions that we saw initially, and we'll briefly talk regarding the right answer. The first question was regarding the study design, where we are following a group of population with an event for the presence or absence of risk factors. And these were the options. The correct answer for this, as we have already seen in our talks, is it's basically a case control study. If you are starting from the presence of risk factors and then following those patients on a long-term basis till the event, then it's a cohort study, but this is the case control study. Question number 2 was regarding the randomized mission, and these were the options. The correct answer to this is option number B. Each participant has equal and known chance of being assigned to different treatment groups. This is what simple random sampling is. When you talk about clustering the patients in age, gender, ethnic, that is basically cluster sampling. Question number 3 was regarding the abstract of the study. What is included and what is not included. All of these options are true, except the option number A, because there are many journals where the abstract is not structured like objective, methodology, results, and conclusion, and you can just give a paragraph. So that is not mandatory, but important results, messages, that should be included. And this is a fact that majority of the people are going to read the abstract. So it needs to be very comprehensive, and it should not be missing any important message and the results of your study. And different journals have different rules regarding the word count that needs to be complied. So before starting writing a paper, writing an abstract, we must see how many, what is the limit for words. It is maybe 200, 250, 300. This is the general range for. Question number 4 was regarding statistical analysis used to compare frequencies. And we had the presentation by Dr. Khalid. And now we know that it is basically chi-square test, which is used for comparison of frequencies. Non-parametric statistical analysis. Non-parametric analysis are basically used for the data which is not normally distributed. If the data is skewed toward one direction, then you can't apply a parametric test like chi-square and t-test and all those. Then you have to use non-parametric tests. Question number 6 was regarding impact factor of the journal. I briefly answered this in initial introductory part. It is basically it's dividing total number of citations of the article published in last 2 years by the total number of articles published in those 2 years. This is what is impact factor. The other, the option A, B, and C, these are in fact other tools of assessing or judging the quality of a journal, like calculating scholar impact based on online research output, news media, and online references. This is basically what H index is all about. Similarly, numeric value indicating the average number of weighted citations. Option A, this is what is IGR, journal ranking tool, another tool which is used for ranking of journals. So as a researcher, we need to know which are the different tools which are used to judge the quality of the journals. But still, the impact factor remains, despite so many limitations and debate about its utility, remains the best known tool for judging the quality of journals and its publications. We had a full session on critical appraisal, and now we know it's basically a systematic process of examining research evidence to evaluate its value and trustworthiness in a particular context. That's analysis combining multiple scientific studies. We know it's basically meta-analysis or review articles. Then evaluation of paper before publication, that's a peer review. And the last question was, which type of research is pre-appraised at the time of publication? We saw in our presentation on critical appraisal that meta-analysis is basically already pre-appraised at the time of publication, while case control, cohort, and randomized control trials, once they are published, then the appraisal can be carried out on these papers. Thank you very much.
So, forum is open for panel discussion and question and answer and feedback. If you have questions, you can write your question in the chat section, or you can raise your hands. So if there are no questions, I would like to end the session, and I would like to congratulate the organizers for organizing such a wonderful research workshop and very nice presentations. Yes,
Excuse me, if I can have few comments about the
Dr.
procedures so far.
Akhtar Shiri want to.
Akhtar Shirin.
Akhtar Shiri, yes.
Yes, if you can give me 5 minutes, I will just want to go through the session so far. The initial paper by presentation by Shahid Sarwar, he had excellent introduction on the what's going to be presented. But I had a comment regarding the impact factor calculations. It was said in the presentation there's 2-year citations. Actually, impact factor is issued for 1 year or 5 years impact factor. For example, if you are having an impact factor of 2019, then we will have citations to the articles published 2 years before that, like 2017 and 2018. So all citations in the 2019 for the articles published in 2017 and 18, and they are divided by the total number of citable items published in the art journal in the 2017-18. So it's a 1 year 2019 impact factor will be calculated based on the citations of the article published previous 2 years. The second presentation, the excellent presentation by Professor Adil Bhatt, and I liked it very much. उसने समंदर को कूजे में ही बंद करने के मुतरादिफ था उनकी presentations. He had given excellent tips for starting off the research presentations and research how to conduct. I can only appreciate a few things I would like to highlight because these are very much needed, especially the training of our researchers for conducting the clinical trials. We are missing the important component is the GCP, Good Clinical Practice certificate for any researcher who want to conduct the clinical research. And there are free trainings available like NIDA is giving, nidatraining.org is giving. It's the National Drug Abuse Center. They are offering free certificate course for GCP. And the second is the clinical trial registration element must be, because if you are going to publish any international journal, they will like to seek the registry number from you. And there are WHO registered the clinical trials registry like clinicaltrials.org and GOV. You have to register it. And the third important factor is that according to the Drug Regulatory Authority of Pakistan, any research conducted in Pakistan must be registered with the DRAP as well. Regarding the Adil Saab's second presentation, manuscript writing, he had given the details of how to write a manuscript, and there he has highlighted few other things which must be emphasized, like the importance of reporting guidelines. Like he mentioned, the consort for the randomized control trial. There are other like STROBE, START, and CARE. And you can all find these guidelines from the Equator Network, which is available on the net free of, you can free access to that. And then he rightly highlighted the importance of selection of the appropriate journal by avoiding the predatory journals. I would like to congratulate him for highlighting these points. Regarding Khalid Mumtaz presentation on statistics, of course, the statistic is very volatile subject, and we can only give orientation during such a short time. However, he has rightly mentioned the overview of the variables and choosing the relevant statistics accordingly. Then the Arshad Kamal Saab presentation on the critical appraisal, especially of the evidence-based medicine. We are living in the era of the evidence-based medicine. He has given the steps of critical appraisal as well as tools required for the critical appraisals. But what I agree with Nateem Tihami Saab, the journal club we are missing. We have to train our trainees to read the article, then critically appraise the article. We should know how the guidelines are made, how to interpret the guidelines, what is the level of evidence, what is the class of recommendation, and how to read a systematic review, a meta-analysis, especially I would recommend that Cochrane reviews must be read by every trainee. And again, in the last Nadeem Tihami Saab letter to editor, it's a sort of post-publication review. And I think everybody should read the articles. And if there is any issue, as he has highlighted, then we should write to the editor, because this will improve the critical skills as well as the writing skills of the trainee. Thank you very much.
Thank you very much, Akhtar Saab. It was a wonderful overview of the whole session. Can we have some comments from Dr. Shahab Abid? Are you there?
Yes, Dr. Altaf, I am here. Well, it's very nicely conducted and a very rich material which has been discussed in the last couple of hours. And I really appreciate the organizer for making such a wonderful arrangement of topics. And the delivery of lecture were excellent. This has been summarized by Akhtar Shireen, so I need not to say anything about it. But my main concern is that the thing which is required first is to have an excellent idea. So until unless you have a good idea, and then you should develop a research question. Discuss and rediscuss the research question. Once you have modified or formalized your research question, only then you can proceed further. The other things will come up later on, that how to do everything. But in order to develop a research question and a research idea, you have to read a lot. What is the existing literature. So that is the main curse of whenever you are doing any research. And think about every paper, any patient which you are seeing as a critical one. So try to catch up whenever you are seeing any patient for a possible case report. My personal experience of publishing a case report is very good. And that's only because I always ask my resident to go and look for every patient and in detail and see if there is anything innovative in those patients or not. For the last 2 years, my group have published 4 papers, 4 research reports in journal like American Journal of Gastroenterology, GUT, and Gastroenterology. The cumulative impact factor is ranging from 12 to to 20. So that can reflect that the case report can be published very easily. Likewise, as the Nadeem Thami says about the critical appraisal for the papers and writing a letter with due respect to the authors, that there should not be anything which is critically against the authors, but you have to express your point of view and your own experience. So these are the two things which will not require much effort. The thing which required effort is a randomized control trial, cross-sectional studies. For these, you need an excellent background reading, and then you have to come up with, your own innovative research idea and to develop a research question. So that's all what I want to say at this point in time. And thank you very much, Dr. Altaf, to engage me in this very useful workshop.
Thank you very much, Dr. Shahab and Dr. Akhtar Sherry. We know you have a very research-oriented background, and you can guide our trainees in, fine-tuning their skills. As chair scientific committee, I thank you all very, very much for being a part of this wonderful session. Over to you, Dr. Nazish, for close. You may close the session now. Thank you very much.
So thank you, Chairs, for your concluding remarks. So I would like to finish this session on research workshop, which is a very useful session. So our next session on acute on chronic liver failure.